Stephan, for the introduction, and thank you to the coordinator for letting me present my work here. And I'm a PhD student, and this PhD project born as a collaboration between the ALD group, formed by Stephan Kemp and Mark Engelen, and the Stem Cell Lab, formed by Vivi Heine. And the title of today's presentation is Human Brain Organoids for adrenoleukodystrophy. And as a brief introduction, adrenoleukodystrophy, it's a disorder which it's a metabolic disorder with a spectrum of pathologies, which main tissues affected are the brain, spinal cord, and adrenal glands. When the brain is mainly affected, is referred as cerebral ALD, and that is characterized by a progressive inflammatory cerebral matter demyelination. When the spinal cord is affected, then it's referred to as adrenal myeloneuropathy, and that is characterized by a slow progressive distal axonopathy with atrophy of the spinal cord and peripheral neuropathy. And it can also be an affection of the adrenal glands, and that means that there is a primary adrenal involvement. A bit on the genetic part, all patients present mutations on the ABCD1 gene, and although that is the case, there is no clear genotype-phenotype correlation, and that is best exemplified by the facts that patients with the most common mutation present all the clinical variants, as well as that monozygotic twins present different phenotypes. And this ABCD1 gene encodes for a protein, a peroxisomal transmembrane protein, which function is to import very long-chain fatty acids into a peroxisome for its degradation. But when mutated, the very long-chain fatty acids accumulate and they are not being degraded. So to study this disorder, many different models have been generated. And recently we went through all the literature in the models, and we published this review that you can have a longer look to it. But today I just want to say that we've learned a lot from them, and they are useful, but each of them only recapitulate one part of the disorder. And in the field, we're also missing the iPSCs. Now we're starting to use the iPSCs to model ALD, but very little work has been done. And the idea with this project is to exploit this technology that I will introduce in a minute to answer some of the questions that we still do not understand. Some of these are why do some people develop one versus the other form of the disease. We also don't fully understand what's the role of these very long-chain fatty acids in the pathogenesis of ALD, or how their accumulation leads to neuronal death and myelin loss. And that's where my research aim comes in, which is to model ALD in vitro using iPSCs to understand the role of very long-chain fatty acids in ALD pathogenesis. But before I'm introducing my setup of a project, I want to briefly mention the Nobel Prize I was given in 2012. So here what you see depicted is the Conrad Waddington diagram, in which a pluripotent stem cell here at the top, hopefully you can see my pointer, through development can either go through different courses to develop and differentiate into different cell types. And this is the picture of mammalian development, and this happens unidirectionally. So the pluripotent stem cell can differentiate into different cell types, but it cannot go the other way back. And the work from Sina Yamanyaka and John Gordon demonstrated that this idea, they changed this idea, in which differentiated cell types can also be reprogrammed into pluripotent cells that can be then differentiated to other cells. And they were awarded a Nobel Prize in 2012 for it. But what does this mean, and why is it relevant for our research? So now introducing the setup, what we do is we have cells from donors and patients. These are usually skin cells or skin fibroblasts. And these fibroblasts are the differentiated cell type that then we can reprogram into induced pluripotent stem cells. So they recover their pluripotent stem cell. They are on top of the hill that I've just shown you. And now through different differentiation protocols, we can differentiate them in 2D to ALD relevant cell types, or in 3D to brain and spinal cord organoids. And although for the 2D, I'm trying to differentiate several cell types today, I will focus my talk on the astrocytes and what are astrocytes in the brain and also why in ALD. In the brain, we have different cell types, mostly known are the neurons, but we also have glia, which were considered just the glue of a brain that would just stick the neurons together. But recently, in the last decade, we've come to know that they have many different functions. And here depicted in blue are the astrocytes, the ones that I'm going to talk today. And some of the main functions that they have in the brain involve the regulation of the neuronal communication. They're also very important in maintaining energy homeostasis. They also modulate the blood-brain barrier, and they are also involved in myelination. And also why in ALD? There was a paper in 2010 from Gort et al., in which they showed that there is an early impact upon astrocytes, upon the cell type in the prelesional areas of ALD patients, in the brain of ALD patients. So what we do is from these skin fibroblasts, we make the induced pluripotent stem cells, and then these ones we can differentiate into these mature cell types, the astrocytes that we have in the brain. That differentiation takes about 90 days. And what we do afterwards is with different cellular markers. Here you see an immunostaining for different markers. We confirm the expression of these markers that then tell us that these indeed are astrocytes. So for example, here you have depicted GFAP, SOX9, C44, ID3, and nestin. So we generated astrocytes from the cohort of patients that we had available, as well as from 3 donors. And we wonder if they would also recapitulate the metabolic deficiencies in patients. So to do so, we measure the levels of the biomarker used on ALD, that is C26 lyso-PC. And here in the graph, you can see here are the 3 controls that I use in my study. These are 4 different ALD lines. So these are 4 different ALD astrocytes in this case. And as you can see, compared to controls, all of them accumulate more fatty acids. And if we keep them longer in culture, please be aware of the difference in the y-axis. This accumulation increases through time. So these tell us different things. One of it is that the astrocytes in vitro generated from iPSCs do generate these very long-chain fatty acids, that they are unable to metabolize them. And this is seen by their accumulation with the graph that I've just shown you, and that through time, this accumulation is increased. And if we then take the fibroblasts from these patients, so the differentiated cell type before was reprogrammed, and also measure the lyso-PC levels, what we observe is that there is no difference between the group of cell types, of cell lines, sorry. However, we do see differences in the differentiated astrocytes. So this is very promising because this gives us hope in the sense that we are pointing in the right direction, and that studying ALD relevant cell types such as astrocytes makes a lot of sense. And currently, to further get deep into the lipid profiles of these cells, we are running some lipidomic experiment, but I still don't have the data, and hopefully that comes soon. But we wanted to check because there was a recent publication in which by increasing the expression of SCD1, which is an enzyme that mediates the transformation from saturated to monounsaturated fatty acids. So saturated are toxic, and these are the ones that accumulate in ALD, and then monounsaturation have been shown to not be toxic. So through a drug that increases the expression of this enzyme, they will see this shift, and therefore the reduction in the free energy fatty acid, the saturated ones. And we wanted to test that in our in vitro model. And what we see is that after a week of incubation with this compound, in this case TO, in all the lines, the levels of the very long-chain fatty acids were reduced. So these tell us as well that, first of all, we recapitulate biochemical deficiencies in patients, and that it also gives us a platform to test different drugs and see how these levels are affected. So as a summary on the astrocyte story, now we've been able to generate astrocytes from iPSCs derived from ALD patients. We are working on the metabolic characterization, and now we've completed the accumulation of very long-chain fatty acids and currently studying the very long-chain fatty acid homeostasis. We've done some work on the metabolic rerouting with the graph that you have here in the slide. And we are running as well lipidomics and metabolomics to further understand the lipid profile and the disease mechanisms. And I'm also as well developing an assay in which we can have a co-culture of astrocytes and neurons to see how astrocytes support the neurons in this case, and if there is any deficiency, any functional deficiency there. So that was for 2D, but these induced pluripotent stem cells can also be differentiated in 3D to make brain and spinal cord organoids. And an organoid is it's a self-pattern, or not self-pattern, but self-structure that resembles an organ, or at least certain aspects of an organ. And this can be derived from iPSCs. This differentiation lasts around 120 days or more. And depending on the patterning, it can go from one or another region of the CNS. And at the macroscopic level, this is a picture of them. This is a plate that I usually culture them. And here you can see the tiny little balls. These are the organoids. And at the macroscopic level, either brain or spinal cord, they look the same. But there's a big difference at the microscopic level. So the cell types that they contain. So first I'm going to talk about brain organoids. But before showing you how they look inside, I want to quickly recapitulate what happens during corticogenesis. So early in development, what we mostly have is progenitors, and these progenitors, called radioglaya cells, amplify the progenitor pool by dividing symmetrically. And around 12 weeks during gestation, they start to divide asymmetrically to produce neurons that then migrate out and specify and form the so-called cortical plates. They also produce some intermediate progenitors in the way that also help in the amplification and the generation of these neurons. So now I'm going to show you some pictures of the organoids in which what we did basically was to fix the organoid so that the tissue inside is preserved, and then we cut it, and then we stain it with different antibodies to see the expression, or the detection of different markers. So for example, here, what you see in red is the labeling of progenitors labeled by SOX2, forming a so-called ventricle-like zone surrounding the lumen, and also some dividing cells here depicted in green. And this region is separated from the neuronal region. So these are progenitors, and these are neurons labeled by MAP2, which is a dendritic marker which labels neurons. And if we zoom in into these ventricle-like zones, we see that they also express GFAP. We see that we see the detection of GFAP, and we see in a way recapitulating how radioglia are looking in vivo. So the cell body is close to the lumen of a ventricle, and then there is this glia fiber that the neurons migrate to the cortical plate. Then if we keep them longer in culture, these progenitors make the neurons. In this case, they are CTEP2 positive, and that means that their identity is of a deep layer of a cortical plate. And there is also presence of TBR2 intermediate progenitors. If we keep them even longer in culture, what we also obtain is not just a deeper layer of CTOF2 positive neurons, but also the upper layer of the neurons, in this case labeled by SAPB2. So this means that we get inside out formation as happens during in vitro development. And if we keep even longer in culture, we get some glia cells that I mentioned before, like for example, oligodendrocytes here labeled by MVP. So that was for the brain organoids, and as for the spinal organoids, this is a set-to representation of a spinal cord during development, and what we see is that there is dorsal and a ventral distinction, and within this structure, different transcriptional factors are expressed, and what does their expression demarcates different domains within the spinal cord of progenitors. And same as before, if we fix, cut, and stain these organoids, what we see is that we have a variety of dorsal and ventral progenital areas, here labeled by PAC6, NKS6.1, PAC7, and OLEC2. And these progenital domains, later on during development, generate different types of neurons. And so far, we have been able to identify motor neurons, here labeled by HB9 ISO1, as well as glutamatergic and GABAergic neurons, labeled by FIGLUT and FIGAT. And if we keep them longer in culture, we also have astrocytes, here labeled by TFAP, and oligodendrocytes by MVP. So far, we have been able to generate iPS-derived brain and spinal cord organoids. The bases that I've just shown you were from donors, so healthy individuals, but now I'm planning to repeat these protocols, but with the ALD lines as well, to then compare ALD versus healthy individuals. And on the plans is to do lipidomics and metabolomics, as well as RNA-seq for the disease understanding part. And then we're also going to characterize the tissue to see if we can recapitulate the disease pathology seen in patients, as well as the Verloncin fatty acid homeostasis study. And as a broad picture of the work that I'm involved at Stephan's lab, is that we're trying to model indeed ALD in vitro to understand the role of the Verloncin fatty acids in ALD. And we do so by metabolically characterizing these cells in organoids. And we're also working on more of a functional or physiological relevant assays to understand the neuropathology. And to do so, we use different model systems. So for example, today I've mainly focused on the astrocyte, but we're also working on making neurons of oligodendrocytes. And these are monocultures, so there is only one cell type in the dish. But then for the neuropathology characterization, we're also working on having co-cultures, so different cell types together in one dish, so you can mix and match and dissect what cell type is the main affected cell type or the main driver of the disease. And then for both things, we're also studying it with organoids. With this, I would like to conclude, and I would like to thank the stem cell lab, as well as the ALD group, and I'm happy to take any questions. Thank you for listening.
Roberto, in the Q&A, there is a question. Do you see it?
Yes, I see a question. I'm just going to read it so everyone is in the same line. It's saying, if I'm using isogenic controls, the answer is no. In this case, I'm getting donor cells from healthy individuals, and then I'm comparing them to iPS from ALD patients. So it's not corrected control, but it's just healthy versus ALD.
There's also a question in the, we have chat and we have Q&A. There's a question.
Oh, sorry,
Floor, shall I read out the question otherwise?
Oh yeah, I have it.
Do you see it?
I have it in front of me, yes. Should I read as well? Yeah, I'm just gonna go ahead. Florian says, I wonder whether organoids can also model blood-brain barrier aspects. Can your system do that? That would be great. But not so far. So in the organoid field, vascularization indeed is one of the things that is still a challenge. And although some protocols are now popping up, which are trying to indeed have this blood-brain barrier integration in the organoids, this is still not really standardized and not ready to go, I would say, for modeling disease. And then there's another question, if there are applications to embryogenesis. I'm going to guess that this is referring to the organoid culture, and on embryogenesis, yes, but that's not what we're focused on, because we're also trying to have myelination in the organoids to then see what's occurring there, because that's a patient phenotype. But there are indeed some protocols that focus on the really early events that happen during organoid formation, for example, to try to model embryogenesis. Then there's another question saying, how much longer before we can see these used more broadly in the research by others as well? That I honestly don't know. Stephan, I don't know if you have a better idea, but
yeah, it is the plan, of course, that you need to upscale these astrocytes and organoids, and yeah, it is very technical. You really need skills to do that. So you need to have a Roberto and a Vivi to do in a stem cell lab to do this. This is not something that a regular lab can do. So
indeed. I'm going to quickly go through the questions before we run out of time. There's a question from Ingrid saying that read a paper claiming that the actual problem is not the accumulation of very long chain fatty acids, but the lack of metabolite not produced in the peroxisomes, medium chain fatty acids. Comments? Yes, that is an idea, right? I also read it, that indeed it might not be the accumulation of fatty acids, but the lack of the other medium fatty acids that are then, when the very long chain fatty acids are degraded, that is the result. But we don't know. That's yeah, it's just a plausible hypothesis, but still don't know.
Well, we do know that very long chain fatty acids are toxic. So
they are the toxic ones. Yes, that is true.
I'll see an interesting question from Florian asking if you can also make DRG neurons.
Oh, sorry. Can you achieve DRG neuron differentiation? Dorsal root ganglia? No, I don't think there's a protocol to do so. I'm currently making spinal motor neurons, but dorsal root ganglia, not yet. So there are some protocols to indeed have different kind of neurons, but these really specific ones, it's hard to get.
There's a question from Amy Leblanc, whether you, well, the co-culture eventually includes cells from the immune system. Maybe you can say something about microglia cells.
Yeah, that's a good question. Yeah, indeed, I haven't mentioned it in this talk, but we're also working on the integration of microglia into the organoids, and then the co-culture into that will be more feasible. Yes.
Because your organoids do not have microglia cells, because you explained it to me. They cannot. You need a different route to make those.
Exactly. Yeah, the organoids in this case are all ectoderm, and microglia come from another cell fate. And there's a question from Alan, what we're hoping for these organoids to do for AMN and ALD patients, and I think that's a pretty relevant question. So the hope is that we understand the molecular mechanisms that are going on, so then we can better develop a drug treatment for patients later on. But to do so, we do need to understand the full picture.
Can I summarize this? Because you see how many questions there are. So that means that there's a lot of excitement. And the beta challenge now is to keep focused for the moment, because we are running in all kinds of directions with this completely new work. And so there's a danger of doing everything and not achieving anything. So that's currently that's the biggest challenge. So if we have 10 Robertos, we can work on all these questions. But yeah, this is so exciting. I think we have to wrap this up, because 2 minutes ago we said, the host said that it was still 1 minute. I think we have to close the session. Thank you very much, Roberto. And,
oh, thank you very much for having me and for all the nice questions and the attention. Thank you.